So shall I start, sir?
Yes.
Yeah. Very good evening to all of you. Good evening, Dr. Kathleen. Good morning to you, Dr. Kathleen, because it's morning for you. Just I would like to introduce about today's session. Today's session is get organized by Microbiology Society of India under the guidance of our president, Dr. A.M. Deshmukh sir. So, Microbiology Society India is founded in 1994 and from 1994 to till date. We are organizing many more activities for the betterment of our subject, that is microbiology. Our students, our members of society is actively participate and actively given a contribution in the COVID-19 area. They had perform many more activities like hand washing, then create awareness about the antibiotic resistance, then the many more activities in the microbiology. And Dr. Kathleen, I would like to inform you that more than 5,000 members are now as a part of our society. And this is another one platform we have developed, that is a MicroJiviTalks. Micro is small and Jivi means bio. Microbiotalks means. So under this platform we have we are conducting many more activities on the same line. So this is also the one very good. We organize our talk on this occasion, that is the biopharmaceuticals from the natural plants, or as a natural medicine from a plant. So I hope these sessions it will be the great for our audience. So I would like to just invite Dr. Deshmukh sir. Please kindly give a few words about on the occasion of this event. Deshmukh sir, please.
Yeah. Good evening to all of you and good morning to invited speaker. I'm very happy to welcome you all, and really this virtual platform have brought us together. There are no boundaries of distance. We can share the knowledge. And today, world is facing the problem of COVID-19. And your topic is a very excellent, because your topic is fighting COVID-19 with plants. This is a very, very informative and innovative topic. I'm sure all attendees from India, they will enjoy your topic. They will enjoy your lecture. But again, I'm telling, this is not the end of lecture. It is the beginning of interaction. And we'll do many activities even in the future. I'm really happy, and I'm thankful to Dr. Kathleen ma'am, for accepting our invitation, coming over on our platform, and ready to deliver the lecture. Thank you very much. Thank you again. Over to Dr. Dahika.
Yeah, thank you, sir. Thank you once again. Just I would like to introduce Dr. Kathleen. Dr. Kathleen is currently a professor at Cornell University, New York, United States of America. Dr. Kathleen was a visiting professor at University of Toronto in the Department of Cell and Systems Biology in 2017. Dr. Kathleen was the faculty of Toronto also in 2010 in the Department of Virology. Dr. Kathleen was the board of director at the TCSD Foundation from 2010 to 2012, and she was also worked at some university, it is Cornell University in 1999 to 2009, and she is also working right now at the Cornell University. Dr. Kathleen has completed her PhD in virology from University of Toronto. Dr. Kathleen has awarded and honored by the many more organizations in life science biotechnology. Some of the few awards I would like to mention. Dr. Kathleen has awarded and participated in the scientists in a Sci-Line from the American Association for Advancement of Science in 2017, and she explain the science and give expert opinion on the scientific issues to journalists. Dr. Kathleen has actively participated in the Fulbright Canada Research Chair in the Global Food Security in 2017. Dr. Kathleen is very active and she has written many more articles on the biopharmaceuticals from the plant. The articles were published in a very national and international journals. One books of Dr. Kathleen is also available on Amazon on the same subject, same title, that is the biopharmaceuticals from plants for the future medicine. So Dr. Kathleen is a very views experience in the plant medicines and how can we develop the different antimicrobials and how can we how can we find out the alternative to the allopathic available antibiotics. So I will not take that much of time. I would like to invite Dr. Kathleen for her lecture. So once again, welcome Dr. Kathleen and over to Dr. Kathleen. Thank you so much.
Okay, thank you. Can you hear me?
Yeah.
Can you hear me?
Yes.
Yeah.
Okay. All right. Thank you. All right. Thank you for that lovely introduction. And yes, I was going to speak today a little bit about fighting COVID-19 with plants. So making vaccines in plants, what's all this about? Well, the basic concept is to produce vaccine proteins in plants which we routinely eat, such as tomatoes, bananas, and potatoes. Oral consumption of vaccine proteins via edible plant tissue has been demonstrated to elicit a robust mucosal immune response in humans. Well, why would we bother to make our vaccines in plants? Well, the World Health Organization estimates that millions of people die, the vast majority of these being children under the age of 5, from preventable infectious diseases. And some traditional vaccines can be expensive and hard to come by. And this making our vaccines in plants can circumvent the need for syringes, refrigeration, trained medical personnel. All of these are major stumbling blocks to providing vaccines to the poor in developing countries. So in case this sounds very much like a sci-fi movie, there are companies that are making pharmaceuticals from plants right now in this fashion. And the first one that was developed is Protolyx, Biofarm Therapeutics. This was this is an Israeli company, and they make glucocerebrosidase, which is an enzyme needed for Gaucher disease. This is a genetic disorder. And they make these in carrot suspension cells. So on the left, you can see there are these very large suspension cells. They're about 8 feet high, each of carrots. And by providing this as a juice, a patient can be provided with enough enzyme to manage their condition daily for pennies a glass. So a regular Gaucher's disease patient who uses a conventional drug, it costs about $100,000 US a year to treat that patient. And this way, by producing the this enzyme in plants, we can break it down to just a few pennies a glass. Another place where plant-made vaccines have been in the news was a few years ago during one of the first outbreaks of Ebola virus in Western Africa. And in this case, we had been designing a antibody cocktail in plants. We started this work using a Gemini virus vector at Cornell University, and then we provided this vector to a company known as MAP Biopharmaceuticals. They produced monoclonal antibodies to block Ebola virus infection using this plant vector to express their antibodies. And they were able to provide this to a few American patients who came back from Western Africa and they had contracted Ebola. So it was given to them as an medicine, and they recovered. So this was one of other noteworthy or newsworthy examples of where plant-made vaccines have been useful. And so I like to think that by making our vaccines and other pharmaceutical proteins in plants, we can make a unique opportunity to impact global health. And on the top here, I'm showing corn. We can actually produce the vaccine protein in corn, provide animal trials and clinical trials, and then completely purify the protein from corn in the form of a regular drug and provide that to developing countries. And that's probably about 1/100th of the cost of a regular conventional drug. Or we can even, what I'm showing on the bottom is we can take a vaccine protein, produce it in an edible plant such as a tomato, and then just lyophilize the tomato so that we can get a powder and then resuspend it as a tomato juice and provide this to developing countries, or the powder, which is quite stable. The vaccine protein is quite stable at room temperature. Or my favorite idea is just to develop the infrastructure and the technology so that developing countries can use their own, build their own pharmaceutical companies that face or address their own diseases, which may not get so much recognition in Western countries, but are still a serious problem in their own local areas. So that's where I'd like to go in eventually with this. So as far as coronavirus is concerned, ironically enough, a few years ago, I think it was maybe 2018 or 17, I received contacts from some Egyptian scientists who are very interested in finding a collaborator to make a plant-based solution to MERS coronavirus. So in these days, this was a real concern for people in the Middle East. This is Middle Eastern Respiratory Syndrome, which is a coronavirus that you may remember. It has a high mortality rate, and it's endemic among camels. And these maps I'm showing you are just indicating where camel culture and camel trading routes are. And they're just to provide some idea of how extensive camels are used across the world. And if you measure up from Egypt and parts of Africa all the way to China, and think of the population of humans who lives in this area, there's probably several billion people who live in this region, and many of those are camels are around, or there's a camel culture where people will not be so happy to be separated from their camels. So it's a real concern that somehow this Middle Eastern Respiratory Syndrome coronavirus could cross the barrier and somehow mutate in such a way that it could become a serious worry for the rest of the world. And so I traveled to Egypt, and actually the picture on the top left is just somebody's backyard, just showing you that camels are everywhere in Egypt, and I'm sure they're in many places in India as well. And I started to work with a collaborator at the University of Toronto. This is Sachdev Sidhu, and I don't think I've ever asked him if his origins are from India. But he's at the University of Toronto, and he had found a way to develop an antiviral protein that could block MERS infection. And the way he does this is he made a synthetic small protein, which on the bottom left I'm showing you in blue. And it binds very tightly. So it's a small protein that he synthesized, and it binds very tightly to the proteinase of the MERS coronavirus. And it won't it prevents the coronavirus from being able to go through its replication cycle. So we wanted to fuse this antiviral protein to the surface of virus-like particles. These are plant virus-like particles, which I'm showing you in the bottom middle. And then we can actually purify these particles and provide them as an inhaler, with the idea that perhaps if someone caught this disease, they could, this could alleviate some of the symptoms, or perhaps their family members might like to have some kind of, way of protecting themselves if somebody came down with this disease. And so for virus-like particles, maybe I'm not, for this group, I'm not sure if probably I'm telling you things you already know, but I've just added an extra slide here. Here I have influenza virus and a virus-like particle, and you can see that the main difference is that there's no nucleic acid in the virus-like particle. So they're non-infectious, yet all of the epitopes and antigens that your immune system would like to see, such as hemagglutinin and neuraminidase, are still available. And in the plant world, we can make these as well. And on the bottom here, I'm showing you Cowpea mosaic virus, which is a plant virus, and we can make empty virus-like particles as well. And we can actually engineer these empty virus-like particles to produce these spike proteins or hemagglutinin, or whichever antigen we're interested in addressing on the surface of these particles, and they're highly immunogenic. And then here I'm just showing you another plant virus called potato virus X. It's a Potex virus, and it actually requires some nucleic acid to become to assemble. But we call these virus nanoparticles, and they still work quite well. So I'm just giving you some examples here of virus-like and virus nanoparticles. So we set about constructing an expression system based on Potex virus, which is something that I had working in the lab at University of Toronto. So on the top here, I have the genomic organization of Potex virus in general. We wanted to take the coat protein and put it into a plasmid expression vector. So this PET29B is a very popular one, and it's also was available with the research labs I was working with in Egypt. So I wanted to make sure I wasn't using something that they couldn't work with themselves, and they could just follow me from where they were. And then we have fused to the N-terminus of this coat protein, we have this Middle Eastern Respiratory Syndrome antiviral peptide, which is a little under 90 amino acids long. It's not a very big peptide. And then there's the T7 promoter, which just turns this protein to be expressed in bacteria. And so here we're showing an electron micrograph. On the left, you can see wild-type virus, and on the right, you can see our virus nanoparticles that we produced in E. coli. And so they're shorter because they're only we're only expressing the coat protein RNA that I showed you in the previous slide. So they are shorter, but they were quite immunogenic. The good thing about this is, although we're working with a plant virus, so far we're only working here with E. coli cells and systems where someone who doesn't necessarily have a plant background can still work with this plant virus and make nanoparticles and make interesting things for biopharmaceutical development with plant viruses. So then we fused, this is our fusion protein here, and you can see, I think, in these lanes, it's a Kamasi gel on the left and a Western blot on the right. And you can see that we were able to make our fusion protein, and it was able to light up with antibodies. And so at this point, we were still working with Middle Eastern Respiratory Syndrome and moving forward in this direction. But we had the situation of COVID-19 crossing the world. And I actually had to leave Toronto and come back to New York because the border closed here between Canada and the US. And it's remained closed for over a year now. Both universities actually shut right down. And New York, you might remember, was one of the initial sites of COVID-19 infection, and it was in terrible shape for several months. So we were not even allowed on Cornell campus for about 3-4 months. And then things were actually quite quiet up in Toronto meanwhile. And right now, the situation has reversed where because the U.S. Because the U.S. has managed to get a lot of access to vaccines, things are opening quickly here at Cornell. And meanwhile, up in Toronto, University of Toronto remains pretty much shut down, and the city is still, slowly getting a vaccine, but the cases are much higher in Toronto and in Canada right now. So based on this, a number of other groups, including myself, started to think about using plants to fight COVID-19. And, it's ironic, again, that I had done all this research with Middle Eastern respiratory syndrome and kind of had was ready to step up for just a slightly different virus. So here are some examples of companies that have used plants to fight COVID-19. The first is Medicago. This is a company that's in Quebec, Canada, and they make virus-like particles of COVID-19. They were able to set these up 20 days after obtaining the sequence, once the sequence was released for the world to see. And they do all of this in plants, and they're on their stage 3 clinical trials right now. So hopefully within a short period of time, there will be many, many vaccines from this group available. I think they're planning for 80 million this year, and then as many as 1 billion by 2023 is what I read. So they're really moving forward with this research. Another group is at University of Western Ontario, and they are making kits using algae. So they're making test kits for COVID-19. And again, this is cheap and easy to work with. British American Tobacco is another company. So I guess they're a UK company that works with Kentucky Bioprocessing, which is a very large company as well, and they're producing vaccines to COVID-19. I think they'll be available after Medicago, but there'll be many available. I know that speaking to the president of that company a few weeks ago, they're spending all of their time making vaccines. And here is Cape Town Biofarms, which is a South African company. They're making a COVID-19 vaccine as well as test kits. And then here's another group at the University of California in San Diego, and they're also using a plant virus to make a COVID-19 vaccine. And here you can see they're using empty virus particles from a plant virus, and they've got the spike protein of COVID-19 attached, fused to the surface. And then they apply these. These can be stored at room temperature. They don't require any cold chain at all. And these can be applied as a patch, and it's just a subdermal inoculation. So it's pretty exciting research, I think, and I hope it is available as soon as possible. So this is a slide that's just kind of a summary of some of the work that's going on in the plant-made pharmaceutical field. So you can see that, I guess at the top right, we have neutralizing antibodies that are being created in plants. And at the bottom right, we actually have what we call neutralizing lectins, but there are plants compounds such as Griffithson, which comes from an algae or a seaweed, I believe, and it actually blocks transmission of COVID-19. So that's being produced. That's on the right. On the left-hand side, you can see that people are certainly making virus-like particles with the spike protein of COVID-19 and testing these out. And that's when I was talking about Medicago. And also Kentucky Bioprocessing are doing these sorts of things. So lots of vaccines and pharmaceuticals are coming available. now that I've kind of covered sort of a general idea for you of what's coming along, I'm back here to University of Toronto and Dr. Sidhu, and we are asking, well, what can we do with COVID-19? And he says, yes, I'm on it right now. And now I'm going to explain how he actually went about making his synthetic peptide. So this is the coronavirus genomic organization. And if you see that there's a bright blue or kind of purple blue gene here, or open reading frame, called PL-PRO, that's the proteinase of coronavirus. And so this is what he wanted to focus on because he could, it has several functions during infection. And if he could block these functions, the virus isn't going to be able to go anywhere. So here, this proteinase has a ubiquitinase function. So ubiquitin is a protein found in cells that labels unwanted proteins, and sometimes these are poorly folded proteins, sometimes they're viral proteins, and they're brought to the proteasome and degraded. And so some viruses, including coronaviruses, express deubiquitinase activity. So they actually can prevent this ubiquitin from tagging the viral proteins and destroying them. So it's kind of an arsenal against the host defense. So what Professor Sandhu did is he used a phage display library. So he just made many primers that are not that expensive to make, different nucleotide primers. And he made the primers to correspond to the conserved regions between where ubiquitin and deubiquitinase would bind to each other. So he found those areas, and then he looked at the conserved regions, and he started to make different types of primers with different amino acid substitutions or nucleotide substitutions to see if he could find a variant that would bind more tightly than the natural deubiquitinase to ubiquitin. And he did this using this phage library, where he just targets his new deubiquitinase to ubiquitin on the bottom. And then I think he used about 8 different rounds. I know in this diagram I'm showing you 2 or 3, but he was able to use about 8 rounds and identify several candidates. He was then able to look at these candidates that had slight changes in amino acid composition. And then he used a spectrophotometer to look at binding to specific targets, so he could identify which ones bound the most tightly. And then he was able to test these candidates in a cell culture to see whether or not they would block infection. So if he was adding this ubiquitin variant, would it bind to the viral deubiquitinase and stop infection? And he was able to find several. So this is work he did. And now he sent to me, here in the States, he sent 3 different variants. And actually one is to SARS, which, is back from 2003, that coronavirus. And we have MERS, and then he's got one for COVID-19, which I'm moving forward with right now. So, as of, I think, this week, I was just looking at what's going on with wealthy versus less wealthy countries, and when is everyone going to be vaccinated. So on the left-hand side, I'm showing you the timeline for vaccination. And it seems that, the dark blue is late 2021, and the medium blue is 2022. And so we have, it looks like India might be the color of late 2022 to have 60 to 70% of your adult population vaccinated. And then we have, 2023 for poor places like Africa, which is, horrible, and, I'm sad for this sort of thing to be taking place. Meanwhile, on the right-hand side, in the oranges and yellows, we have the daily cases. And so, India is obviously just, we're just reading constantly over here in the US about India and the issues going on with not having enough equipment, and a lot of deaths, and even a black fungus we're reading about. And this is a rare disorder that we're hearing about over here that seems to be becoming a serious problem for patients. It's just horrible and sad. And obviously there's some other countries as well that are still suffering, but it's you're certainly in the news over here all of the time, in case you're wondering. So back to my expression systems. So now that I have this research done, to an extent in plants in kind of a small scale, we have to think about what the next steps are. So I've shown you here a little bit about using what we call transient expressions. So it's just short-term expression in plants to produce virus-like particles or virus nanoparticles. Another step is to use transgenic plants to produce these vaccine proteins or antiviral proteins. And that's certainly of use. It has its advantages and disadvantages. It's slower to produce a transgenic plant, obviously, than to use a plant virus. But at the same time, you can get seeds from transgenic plants. So it has some other advantages as well. And then the next thing is to get these to enter into human cells. And actually, some research has been done to show that some plant viruses actually can enter human cells. And I'm trying to show you here some data from a colleague of mine, but the yellow that you see is inside of cells, human cells. So these are a particular virus, Cowpea mosaic virus, which actually can enter cells. So that's very exciting. They enter through a protein called vimentin. And so it makes them feasible as delivery vehicles, these plant viruses, to get pharmaceuticals into cells. So I've started a collaboration with a company. This company is in Boston, Massachusetts. It's called Zia Biosciences, and they're very interested in setting up research projects with academics who want to produce large quantities of pharmaceutical proteins in plants. And the technology they have is a very fancy greenhouse. That big white block you see is a very fancy growing facility where they can produce plants that grow exponentially, and they have ways of determining which, how to best produce the protein of interest or compound of interest for that matter. So it's, I think it could be very useful for anyone in the kind of field I'm in, which is molecular farming, or else anyone who's interested in plant-based medicines. These folks know how to produce your compound of interest at high levels very quickly. So it's really a very useful technology. This is just more of the same. So it just gives you an idea of how they develop this. And I've also got a project where we're looking at producing just exactly, it's more of a research of where this ubiquitin variant will go and how it will, how well expression, what kind of expression we can get. And this is my Gemini virus system that I showed you before with the Ebola project. And so here I've just fused this ubiquitin variant to a GFP, and we can monitor where it goes in cells. So this is just something we're working on right now. And here I've just kind of put a summary slide of the different systems that I'm looking at for expressing this ubiquitin variant. So we can use transgenic plants, we can use the Gemini virus, we can use suspension cells, and we can make these virus-like particles. And on the bottom, I guess that should be the hours, but basically days. How long does it take to get these proteins? What does it cost? What will be the economic situation? So we're looking at all of these right now and trying to figure out the best way to move forward with these different expression systems. And that's about all I had to say. So just thank you again to University of Toronto and also Cornell University. And I'm going to stop sharing my screen and ready to take any questions.
Yeah. Dr. Deshmukh, sir?
Hopefully it'll get out of...
Any questions you can ask, please.
Can I ask?
Yeah, ma'am. Yeah.
Good morning, ma'am. It's a very interesting talk. I have one question. How can we control the doses if we make the vaccine in the plants?
Yeah, great. So if we can do this quite easily if we just lyophilize plant tissue and quantitate it and then resuspend it. So we can't. The plant will express proteins at variable levels. It's not good enough to, for example, eat a raw banana and think that you'll get enough vaccine protein in you. You probably would have to do something where you either dried the banana up and ground it up and did something like that, I think, because it's just too, there's too much variability in concentration when you're expressing it in plants. Though my group at ZIA, as I mentioned at the end, who I'm collaborating with for, possible, large-scale production, is he seems to have some confidence that he can change that variability, he says, to less than 7%, which is really quite incredible. So he actually might be able to produce this protein without, in a fresh fruit, for example, or vegetable.
Thank you, ma'am.
Yeah.
Okay, I have a question here. Oops, sorry, it just left. You'll have to read it.
Yeah, any questions, please, audience? Jia Muhammad, yeah. Are you online? Jia Muhammad is asking, ma'am, what are the challenges arises during the productions of plant vaccine?
What are the challenges?
Yeah.
Well, I think right now, I would say one of the biggest challenges has just been regulation, because a regulatory framework in the US and Canada didn't really exist. And so I think it's a wonderful idea, and I think it has a lot going for it. But we don't have a solid regulatory framework. And I think as a result, some investors are less likely or hesitant to invest big time in this. And that would be why. So I think we can, it's just a matter of time. And Frank, right now, I did mention Medicago with their phase 3 trial. They might be the first to get their vaccine commercially accepted and move forward. And I think that it will be much easier for everybody else after that happens. So I would say that's probably number one. And number two is to make a lot of vaccine, you need a lot of plants. So we need to have make large manufacturing facilities that are plant-based.
Yeah, thank you, doctor. Next, another question is asked by Dr. Amit Singh. Amit Singh, are you online?
Yes, sir.
Yeah. You please introduce yourself and you can ask questions.
Good morning, ma'am. I'm Amit from Faridabad. I'm doing my MSc. Ma'am, can we get hybrid plants using the seeds of vaccinated plants in next generations?
Yeah, I think you can backcross them and produce plants with these traits and save them, and then just grow them when you need them. Yes, that can be done. So the answer is yes.
Okay, thank you, ma'am.
Dr. Vandana Sharma. Dr. Vandana Sharma, are you online?
Yes, sir.
Yeah, please introduce yourself and from which institute you are, and please ask your question.
Good morning, sir. Good evening, sir, and good morning, Kathleen, ma'am. I'm working as faculty in Department of Microbiology in DSPW University, Ranchi. Ma'am, I have one question that in the current situation is as everybody knows that current situation is very bad in case of COVID. So how much time it will take to develop a vaccine to come into the market?
Okay, it's great. So, we have, I guess there's a lot of conventional vaccines, which, hopefully you're going to see that aren't necessarily plant-based. I think one of the questions that I had through my reading is, we have an abundance of vaccines in the U.S. right now. So we have more vaccines than we have people, I think. And I thought that perhaps some of these vaccines might be hoarded or kept here because people would be concerned about having to get a third booster, for example, because we weren't sure how the immune system would work. Though I have been reading recently that the immune system, because there's a response, the immune system will not require a booster for quite some time. So that's good news for distributing vaccines to others. And, the inequity is horrific, isn't it? So with the plant-based vaccines, I think that Medicago will, within a matter of months, be able to provide, start to provide. They're fast-tracked to provide vaccines. They're on their stage 3 clinical trials. There are more than enough participants in these trials because there's so many cases. So I would imagine within 2 or 3 months, those trials might be complete. And there's no adverse effects. They're safe and easy and efficacious. And then it would be fast-tracked by the FDA in the US to become a vaccine. And then it'll be very quick to make a lot. So I would think that you'll start to see or hear about a lot of them becoming available this year. And I wish I could give you more firm timing, but it's probably, that's, I would think within 6 months, you might start hearing about them. So hopefully we'll get them over to you.
Thank you so much, ma'am.
Yes.
Ma'am, one more question. Can we mix the plant-based vaccine with the animal-based vaccines?
I don't see why not. I think sometimes there's a good reason to have a sort of a heterogeneous prime and boost. So, yeah, I would think there's no reason to say no to that. And again, this vaccine would be based on plant virus-like particles, and they would be stable at room temperature. So that they should really be focusing on countries where refrigeration can be a problem. I think that's where they need to go.
Thank you so much, ma'am.
Yeah. Dr. Sanu Divakar from Central University of Punjab. Are you online?
Yes, sir. Good morning, ma'am.
Yeah.
And as you presented very well. And I am also interested in phytochemicals-based drug, and I have also done in this field using some immunomodulatory compounds against COVID. So I want to know about that. Is there any specific phytochemicals which we can use as a plant vaccine against COVID?
Yeah, one that I hear that is having a lot of success in clinical trials in a U.S. hospital is this Griffithson, which I think is a plant lectin, but I think it does come from, I think it's red algae, but it's looking great. It's also it prevents infection from several viruses, not just COVID-19. So it must be sticky on the spike proteins of certain viruses. Now, I do have a list of phytochemicals. If you email me, I can send you my paper, or you can Google and find it. And it would have, there's a nice table about plant phytochemicals. So for
Okay,
COVID-19.
thank you so much, ma'am.
Yeah.
It means a lot for me, ma'am. I'll send you.
Yeah, Mukesh Ramakrishnan. Next question is by Mukesh Ramakrishnan.
Hello, ma'am.
Yeah, introduce yourself. Yeah.
Yes, ma'am. I'm Mukesh, and my question is very simple. And we still don't have a vaccine for treating common cold. Can we expect to have a vaccine to treat common cold, which is caused by the rhinovirus? And one more question. Can we use the plants which are grown through the method of hydroponics to create vaccine? Will it be effective? That's my question.
So for the second part, you're mentioning hydroponics. Can we use hydroponics?
Plants, can we use plants which are grown through hydroponics for the purpose of production of vaccine?
Yes. So that answer is certainly yes. There, one of the big companies in the States, it's called iBio, and it's in Texas. And they use all hydroponics to produce vaccines and plants. And they're also making a COVID vaccine, but they're not as far ahead as Medicago is. So they're doing the same thing, and they use hydroponics. So yes, it's actually, in certain cases, it's preferred because you get this homogeneity of expression. I think certain plant platforms have their advantages, and that's one thing I've heard. As far as making a vaccine to the common cold, yeah, I don't see why we can't. I think you're not talking about the flu, you're talking about the cold. Yeah, I think that I'm not actually sure why efforts aren't made to do that. Maybe they felt, vaccine companies felt it was not the low-hanging fruit or something. But I see no reason why not, put it that way.
Maybe it's not as dangerous as the other viruses.
Maybe it's not as dangerous, yeah. It would be nice, though.
Yeah, come to Dr. Deshmukh, sir. Any comments, please? Deshmukh, sir.
Yes. Now you are concluding the session. Correct?
Yeah, yes, sir, yeah.
Yes.
So before that, again, I want to express my thanks to Kathleen ma'am for delivering a very, very excellent lecture. It is a new aspect, that is how to use plants to fight against COVID. And that's why many research workers today, they have joined. This is a new area, new theme. And I will request to all attendees that if you want email ID of ma'am, you write either in the group or on personal chat of Dr. Dahikar or myself. We will give email ID to you. That was our main intention, to increase the communication in between scientists from US and scientists from India. So Kathleen ma'am, you are the beginning. You are the beginner. Now many more scientists.
From Nepal too, sir.
Yes.
We are also ready for Nepal too.
Okay, very nice, very nice. I will introduce Kathleen ma'am. He is the head of department from Kathmandu, Nepal.
So please do.
So they will also invite you because the students in his college, DAV College, Nepal, students are very, very active. They will be happy to listen you also.
Definitely.
Yes. So he is a Professor Shashi Bhushan from Nepal. So I am very happy to welcome all delegates from India, Nepal, and today really on this platform we are together from different countries, but discussing about the microbiology. Always I am telling microbiology is the science needed for existence of human being. And we are the students of microbiology. So thanks again, Kathleen ma'am. We will again keep in touch with you and we will work together, we will interact together even in the future. I am thankful to Dr. Dahikar also for arranging such a beautiful lecture within a short time notice. So thank you again. Thank you everyone.
Yeah, thank you so much, sir. Thank you. Thank you, Deshmukh sir, for your kind words and motivation to all microbiologists. And definitely, the scientists from India and will be connected with the Dr. Kathleen. And Kathleen is very cooperative and she has accepted our invitations within a very short period of time. Because day before yesterday, I had called her through email and she just accepted. Yes, I am ready to deliver a talk. So really, thanks, Dr. Kathleen, for your nice talk on the biopharmaceutical, and definitely we will be connected for the further research collaborations. So once again, thanks to all. Thanks, Dr. Kathleen. Thanks to Dr. M. Deshmukh sir and official permission of Dr. Deshmukh sir and our participants. I'm concluding today's sessions. So have a good day and take care. Bye bye.
Bye bye. Thank you.